Hello friends, good morning to one and all, and I am here Vandana Kushaha to discuss today's topic, that is nucleic acids component. We are going to discuss today nucleic acids and nucleic acids components. Okay, what are the nucleic acids? Nucleic acids are the micromolecules store the genetic information and enable protein production. Friends, if we will classify the biomolecules which are found in the living organism, then we can classify in the two categories. First one is the micromolecules, and second one is the macromolecules. What are the micromolecules? They are very small in the size, like vitamins, water, minerals. And what are the macromolecules? They are large in size, like carbohydrates, protein, as well as the DNA and RNA. Here it is written nucleic acids are micromolecules that store the genetic information and enable the protein production. Yes, nucleic acids are the actually molecules which enable the protein production. How they are producing proteins, we will see in the next session, because it has the long process. First, it will be go for the nucleic acid replication, then we will go for the nucleic acid transcription, then after that the translation, then it will go to the further protein products, and then it produce the proteins. It includes the DNA as well as the RNA. Nucleic acid was the first discovered by the Friedrich Mischer from the nuclei of the pustles leukocytes from the discarded surgical bandage and called it nucleins. Means when it was the first discovered, they called it name as the nuclein. Okay. These are the naturally occurring chemical compounds that is capable of being broken down to yield phosphoric acid, sugars, and mixture of the organic bases, including purines as well as the pyrimidines. Friends, if we will do the breakdown of the nucleic acid, then we can get the three components, which is the written here: phosphoric acid and sugars and mixture of the organic bases. In organic bases, we can classify the two types, that is purines as well as the pyrimidines. We will discuss in the next class slides. Two main classes of the nucleic acid: deoxyribonucleic acid as well as the ribonucleic acid. These are the called as the biochemical basis of the heredity. Friends, these both micromolecules are called biochemical basis of heredity because they are responsible to store the genetic information, and they are responsible to transfer the gene from the one generation to next generation. Here we are going to discuss the components of the nucleic acids, that is the nucleotides. First we are going to discuss the nucleotides. The monomeric units of the nucleic acids are called nucleotides. Therefore, these are called polynucleotides. Nucleotides are phosphate esters of the nucleosides and made up of the three components. First is the base, that has the nitrogen atom, nitrogen base, and second one is the 5-carbon sugar, and third one is the and ion of the phosphoric acid. We have concluded that means nucleotides is made up of three components. First one is the nitrogen base, second one is the sugar, and third one is the phosphoric acid. Okay, we will discuss all the components one by one, and here we will going to discuss nitrogen bases. What are the nitrogen bases? Nitrogen bases are the heterocyclic, planar, and relatively water insoluble aromatic molecules. Means heterocyclic means a cycle have the different atoms like carbon, hydrogen, oxygen, nitrogen, and it has the cyclic, that is ring structure, and it has the planar structure. Means if we will form the structure, then it will be all atoms will be lying on the same plane. And relatively water insoluble, actually they are soluble in the organic solvent like ether, alcohol, and others, and are aromatic molecules because of the ring structure. There are the two general types of the nitrogen bases in the both DNA as well as in the RNA, that is called purines. These are called pyrimidines as well as the purines. Okay, we can see here the purines structures are here. Two different nitrogen bases with the purine ring composed of the carbon and the nitrogen are found in the DNA. Two common purine bases found in the DNA and RNA are the adenine Adenine is called 6-amino purine. Okay. 6-amino purine. We can see here the structure of the adenine, and we are counting. Start from here. We are starting counting from here. 1, 2, 3, 4, 5, 6, and here in sixth position is the attachment of the amino group. That's why we are calling 6-amino purine. Okay, and except this amino group, we can call it is the purine ring. Okay, that is purine structure. And here it is guanine structure. Except external groups, like here in sixth position carbonyl groups, and in the second position amino group, this except these two external groups, we can call it the purine ring structure. So it is called as the 6 oxy 2 amino purine. It is called as the guanine. Okay, next we will discuss here pyrimidine. What are the pyrimidine? What are the bases in the pyrimidine? Two major pyrimidine bases found in the DNA. First is the thymine and cytosine, as well as the uracil. Thymine and cytosine in the DNA. Uracil is found in the RNA. Thymine is also called 5-methyl-2,4-dioxypyrimidine. Okay, this is the thymine structure. Here we can see, and we can start the counting from the this below side nitrogen, and we will count in the anti-clockwise structure. This is the first, and this is the second. In second and fourth group there is the oxygen group. This means this is the carbonyl group, and thymine is also called 5-methyl 2,4-dioxypyrimidine. That's why, because of at the fifth place of the thymine methyl group is the present, so it is called as the 5-methyl 2,4-dioxypyrimidine. And here it is, we can see the structure of the cytosine 2-oxy-4-aminopyrimidine, and in the uracil it is called as the 2,4-dioxypyrimidine structure. Okay, thymine contains the methyl groups at the C5 position with the carbonyl groups at the C4 and C2 position. Already I have told you, in the C4 position and C2 position, carbonyl group is present in the thymine. Next slides we are going to discuss about the sugars. What are the sugars present in the DNA as well as the RNA? Naturally occurring nucleic acids have the two types of the pentose sugar, ribose sugar as well as the deoxyribose sugar. Ribose sugar is found in the RNA, and it is 5-carbon monosaccharide with a hydroxyl group on each carbon. See friends, here it is the ribose structure and here it is the deoxyribose structure. We will start counting from this topmost position, and one first carbon is there, second and third, fifth, fourth and fifth carbon is there. This is the CH2OH. Okay, we can see here in the deoxyribose sugar, in the 2' dash, in 2' there is a absence of oxygen, means there is a removal of oxygen, that is deoxy, and here is the oxygen is the present, means hydroxyl group is the present. Deoxyribose sugar is found in the DNA. It is 5-carbon monosaccharide lacking one oxygen at the 2' position. The hydroxyl group at the 2' position of the ribose sugar is replaced by the hydrogen. Okay, I think you are getting my point. Even if you are not getting my point, if you are not understanding anything, you can comment in the below in the comment box. Then I will reply. Yes, sir. What is the sugar puckering? This sugar puckering is very important in the DNA structure as well as the RNA structure. Pentose sugar is the non-planar structure. This non-planarity is termed as the puckering. Because we know that the pentose sugar is the non-planar structure. Due to that, due to non-planarity of the sugar structure, this process is occurring. It is called as the puckering. Pentose ring can be puckered in the two basic conformation. First one is the envelope and second one the twisted. What is the envelope form and what are the twisted form? We will say here. In the envelope form, 4 carbons of the pentose are nearly coplanar and fifth is away from the plane. Okay, means in the envelope form what happens? 4 carbons in the same plane and other will be that fifth atom will be away from the plane. In the twisted form, 3 atoms are the coplanar and other 2 atoms lie away on the opposite sides of the plane. Twisting the C2' and C3' carbons related to the other atoms resulted in the twisted forms of the sugar ring. Purines show preference for the C2' endo and pucker conformational type, whereas the pyrimidines favor the C3' endo. Friends, here we are saying that preferences of the structure means someone is stable in them. Means purines are pyrimidines they are establishing their structure in the different forms. If they get stable the structure means they will prefer that structure. It may be endo, it may be exo. Then we can see here, and purines show preference for the C2' endo, pucker conformational type, whereas the pyrimidines favor the C3' endo. In RNA, we find predominantly C3' endo conformation. Here we will discuss about the nucleosides. Friends, don't get confused here. Two terms are there. First one is the nucleosides and second one is the nucleotide. Nucleosides we are going to discuss that is contain sugar as well as the nitrogen bases. Sugar and nitrogen bases join to form the nucleoside. The bond between the sugar and base is called N-Glycosidic bond. Means if there is the presence of the sugar and only nitrogen, then it is called nucleoside. Friends, one thing remember you should remember one thing that here phosphoric acid is absent. Okay, there is no presence of the phosphate group. Nitrogen base lies above the plane of the sugar. When the structure is written in the standard orientation, the configuration of the N-glycosidic linkage will be beta. Okay. Nucleotide. Nucleotides are the phosphoric acid esters of the nucleosides. Already we have discussed the slides. Means already we have discussed the nucleotides, and there are three components are there. First one is the sugar, second one is the nitrogen bases, and third one is the phosphoric group. Means here phosphate group is the present at the 5' position. A nucleotide can therefore be called nucleoside 5' phosphate. A nucleotide can have the 1, 2, or 3 phosphate group at the C5' position and are called nucleoside monophosphate, diphosphate, triphosphate. Polynucleotides are the polymer of the nucleotides which are formed by the condensation of the nucleotides. Polynucleotides are the polymer of the nucleotides which are formed by the condensation of the nucleotides. Condensation reaction occurs between the alcohol of the 5' phosphate of the one nucleotide and 3' hydroxyl of the second with the elimination of the H2O, and this elimination is H2O and formation of the phosphodiester bond. Friends, here we are going to discuss structure of the double stranded DNA, which are made up of the two long polynucleotide strands. First, we will discuss about the different. DNA have the different forms. Watson and Crick first described the structure of the DNA double helix in 1953 using Xray diffraction data of DNA fibres. Friends, X-ray diffraction technique is the molecular biology technique in which we can view the structure of the 3D structure of the DNA as well as the RNA as well as the proteins. Watson and Crick double helix model describe the feature of the V form of the DNA. However, there are the many other forms of the DNA like A, C, Z forms which are very distinct from the V form of the DNA. Here we are going to discuss about the features of the V form of the DNA. Okay, that is the two long polynucleotide strands coiled around the central axis. The strands are wrapped platonimically in the right-handed helix. Friends, there are the two types of the DNA which exist in the nature, like right-handed helix twisted as well as the left-handed twisted. So V form of the DNA is the right-handed helix twisted. Strands are antiparallel, that is one strand is oriented in the 5' to 3' direction, the other in the 3' direction. Yes, because of anti-parallel direction, if one strand will be in the 5' to 3', then must be other strand will be in the 3' to 5' direction. The strands interact by the hydrogen bond between the complementary base pairing. G forms with the 3 hydrogen bonds with the C, and A forms with the 2 hydrogen bond with the thymine. G is the guanine, C is the cytosine, A is the adenine, as well as the T is the thymine. We know already. Okay, continue. Angle of interaction. Yes, we can talk about here, that is major group and minor group formation will occur in the DNA. In the DNA. Angle of interaction between the base pairs result in the major and minor group. Angle of between the C1 prime atoms is larger on the one side than the other, denoting two dissimilar groups in the DNA. Means major and minor group formation will depend on the angle of interaction of the DNA. Okay, the side containing N7 of the purines is termed as the major group, and while the other side containing N3 of the purines is the minor group. If we will talk about the diameter of the helix is the 20 angstrom. Helix rise per base pair is the 3.32 angstrom. We will discuss in the next slides, that is the structure. There are the 10.4 base pair per helical. Base pairs are in the site. Inside, the molecules stacked close to each other. Friends, here we can see the structure on the DNA, that is double stranded helical structure. And we can see here the two strands are there. If one strand will be in one side 3 prime, then next side will be 5 prime. And if one strand is the 5 prime will be there, and the next side will be 3 prime, because it is anti-parallel in the structure. Okay, and we can this is the helix length, that is 34 angstrom, which is the length of the major group to major group or minor group to minor group. And here we can see the 1 ton per base pair rise, that is 3.4 angstrom, and here attachment of the phosphate deoxyribose sugar, which is the vertical bars, and phosphate deoxyribose sugar is the backbone of the DNA. You should know. Nitrogen bases are present in here. These are the vertical bars, which is the form of the nitrogen bases, and here A bind with the double bond with the thymine, and a G bond with the G makes triple bonds with the C to form the guanine and cytosine base pairs. And we can see here. And friends, if this video is helpful for you, please like, hit button, hit like button, and get subscribe my channel. Thank you and all. Bye bye.